We've been focusing on trying to understand what are the what are called antigens. What are the antigens that are recruiting T cells into the tumor? The essence of immunotherapy is to activate T cell killing in a tumor, and there has to be something to draw them in, and once they're drawn in, things like checkpoint inhibitors can be activated. And something that's emerged over the past couple of years, this is probably the most rapidly developing field of cancer research that exists, and certainly has existed in my life, is that antigens, which are fragments of proteins that are maybe only 10 or 11 amino acids long, that contain mutations, so they're fragments of mutated proteins, are very, very important for immunotherapy, and largely because they don't look like self, because they have mutations. And so if you can trigger a T cell response that is associated with these mutated antigens, then that T cell response will just affect the tumor, nowhere else. And so we've been developing technologies that allow us on a per patient basis to identify what neoantigens, they're called neo if they have a mutation, what neoantigens are drawing T cells into the tumor, and then what are the functional properties of those T cells once they're drawn into the tumor. It's an interesting concept because the neoantigens themselves are being viewed as potential vaccines, and the T cells that are attracted to them are viewed as candidates for engineering T cell drugs. And the interesting concept behind this is that you actually have to start with the entire genome of the patient. Everybody's different. And so you analyze the patient's genome to identify what are the mutated proteins. You look at the RNA expression data to identify which of those proteins are actually being made in the tumor. And then you take those proteins that are being made and also just computationally, you chop them up as if they were being enzymatically digested. And for all those fragments, then you calculate, once again, it's all in silico, which of those will be fit into what are called major histocompatibility complexes, which is how they're displayed for T cells to see. So you do this all computationally, and you come up with a list of maybe 200 or 100 neoantigens in a given tumor that might be responsible for drawing T cells in. And then you have to test them. And testing them is that's the challenge that we've been developing. We've developed a nanotechnology microfluidics approach that allows us to build basically a test for the entire list. And we find quite a few of them there, and we're able to even follow those. It turns out those T cell populations that recognize those neoantigens are in the tumor, but they also are in the blood. And so we're able to follow the same populations in the blood just over the course of the therapy. So we're looking at patients that are on trials. And what we found, and now these are melanoma patients that are responding pretty well to anti-PD-1 therapies. What we found is that something like half of the what are called CD8 T cells, which are the major effectors, half of those T cell populations are specific to neoantigens. So that's a big number. It tells you overwhelmingly that these neoantigen-specific T cells are what's killing the tumor. Well, it was completely unthinkable a few years ago. Right now, to get a genome sequenced, maybe it's $2,000. In 2 years, there's companies that tell me it'll be down to $400. $400, it almost makes sense for everybody to have their genome sequenced. Our analysis, so for example, we were doing this entire analysis of a large library of neoantigens to try to figure out which ones were important and analyzing the patient's blood. I had a postdoc who started doing an analysis Friday afternoon. I landed here in New Orleans on Saturday morning. I took like an unpleasant red-eye flight, and I had the data. And so the technologies we developed, and that was searching through, on the order of 100 neoantigens. So it's, for sure, it's an expensive thing right now, but it's getting cheaper very, very quickly. Everybody's different. And so the challenge, and you mentioned the cost, the challenge is, can we identify these candidate vaccines or candidate T-cell receptor genes and turn those into a therapy in a meaningful time frame for a patient? And I think I don't see any barrier to doing that at the moment. There's a lot of stuff we have to develop, but I think, one thing associated with technology that people get misled by is that you tend to look at what is the technology now, and then you extrapolate linearly. So if I get cancer in 3 years, am I going to be able to manage this technology? But technology evolves exponentially. And so trying to project where we are now and where you're going in a linear fashion doesn't work. This will be, I would guess that, let's say it costs us $50,000 to do the entire thing, labor, everything for a patient. Now, in a couple of years that might be down to $1,500. And that's very doable. That's not ridiculous. And that's just how technology evolves. So we started working in this field with my colleague Tony Rivas something like 12 years ago. And at the time it was a backwater, and there were hints of success. Steve Rosenberg's group was doing well. We had trials that we were involved in where you had patients respond, but it wasn't durable. And over the past 3 years, of course, it's become the forefront of cancer therapy. And it's just amazing. It's just amazing. And once you have signal, once you have patients that really have durable responses, you can always make it better. And that's, I think, the thrust of the field now is to extend those durable responses to larger and larger patient populations. And I see no reason why that's not going to happen.